DNA to RNA. The second part of the central dogma is translation, and that's where we left off on Friday, right? Going from an RNA to a protein. Right? So we talked a little bit on how to read an RNA, right? How to read codons. We went through, right, this example here, which I told you I inadvertently made it a little trickier, right? I did not put a start codon until the second codon here, which is why we're starting with methionine instead of the amino acid for CCU, right? I don't think I would do that to you on an exam, right? That feels tricky to me, and I don't want to trick you, right? But knowing how to read sets of threes and translating it into an amino acid, great skill to have for an exam. There's likely going to be a question on that. All right, but that's where we left off, right? So this was the next slide that we would have gone to on Friday. So we know how to read the codon. We know how to read our genetic code, right, in our codon translating image here. But we didn't talk about why this genetic code has so many important properties. And this is going to help you answer question number 5 on the content check that we'll be working on today. All right, genetic code, it is redundant, meaning all the amino acids are going to be coded for by more than one codon. All right, so if you look at the top corner there, let's look at the second row under C here, the first row under C, right? We see the amino acid serine. We see that UCU, UCC, UCA, UCG, they're all coding for one amino acid, right? Serine. So serine is coded for by more than one codon, which makes the genetic code redundant. Right? Not only is it redundant, but it's also unambiguous, which means that a single codon is never going to code for more than one amino acid, right? If we're going to look at our example of serine on that top row there, UCU, that codon, always going to code for serine, right? Never going to code for anything else. Right, so it's redundant, it's unambiguous, and it's non-overlapping. And I'm going to draw on the board to help demonstrate this one out here. All right, so non-overlapping. I'm going to make up a sequence on the board. We'll do AUG, which is the common start codon. Let's do some A's. All right, if we were to read this sequence of mRNA, right, starting at the 5' ending at the 3' here, I'm always going to read it by sets of threes. So I know AUG is going to be my first one. AAC is going to be my next one, right? And then you'll continue on. I'm going to read AUG, and then I'm going to move on. I am not going to read AUG, and then read UGA, and then GAA, right? That would be overlapping, right? We're never going to overlap our letters, right? The first 3 letters stand for a codon. We're going to move on to the next. That's what it means to be not overlapping, right? So instead of reading AUG, and then UGA, and then GAA, all right? We're just reading it by sets of 3. They're not overlapping, right? Once a letter is used, it's not part of any other codon. The last thing about this genetic code is that it's conservative, which means, well, I found my pointer, but my pointer doesn't want to work now. There we go. Which means that the first 2 bases in a codon is likely going to be the same for amino acid. So again, we're looking at serine up here. The first 2 bases are UC, right? It's the third base that changes, right? So this makes it less likely that if a mutation were to occur in our genetic code, right, say this UCU becomes UCC, that U becomes a C, right, it's still coding for the same amino acid. So even if that mutation were to occur, it's not going to affect the final product, right? So we can see many examples of this in our genetic code here, right? The amino acid for THR here, I forget the full name off the top of my head, but it's conservative in that the first two letters are always AC. Right, so it's redundant, right? There's going to be more than one codon for each amino acid. It's unambiguous, which means a single codon always refers to the same amino acid, right? If it's UCU, it's always going to be serine. It's non-overlapping, right? So we're reading it in sets of 3. Once we have read a letter, right, we're not using that letter in another codon. And it's conservative. We're keeping those first two bases the same in many of the examples in our genetic code. All right. We have talked about how to read mRNA, but not how it's physically assembled. Right? So we've talked about how to go from DNA to RNA and RNA from a protein. But what actually reads that mRNA and builds that protein strand? Right? Well, this is where we have a couple of different molecules that are involved. One being mRNA. Right? This is the one we synthesized in the DNA. It's left the nucleus is into the cytosol here. It's denoted by this green strand. That's our messenger RNA. It contains codons, so sets of 3 RNA nucleotides that we read as a set of 3 down the line. All right? So mRNA we're already familiar with. But the next one is tRNA. Right? tRNA is the one doing the reading. Right? So as we look at a piece of paper and we're translating a codon into an amino acid, right? By us doing that, right? That's basically the role of the tRNA. It reads the codon and it figures out what amino acid goes in next. Right? So tRNA would contain an anticodon. That's how it reads the codons. If we look at this first example here, right, we have a codon of AAA. The tRNA is reading that codon and knows that it has to match up the anticodon that it carries. Right? The anticodon to AAA would be the complement to it, which would be UUU. So the tRNA that has the anticodon of UUU would come in and connect with this codon here, right? And what it brings with it is an amino acid. The amino acid that's coded for by the codon AAA. Right? So tRNA is a molecule that has an anticodon. The anticodon is complementary to the codon. Once we match up the codon to the anticodon, that tRNA molecule is carrying the appropriate amino acid that is called for next in the sequence. All right, we're going to go over this in a few different other ways to help us really understand what's happening here. All right, I like this next slide because it really puts everything together. Transcription and translation. If we look at the top, we have our DNA strand. Right? We know that one strand is going to be transcribed into mRNA. Now we know mRNA can only be transcribed or built in the 5 to 3 direction. So if we're building 5 to 3, we have to read 3 to 5. So we would read this template strand that goes from 3 to 5 to build our mRNA strand from 5 to 3. All right, so that's our first step, transcription. And here's where our tRNA molecules come into play. We go from our mRNA molecule, right, to our line of amino acids. So if we look at the start codon, AUG here, right, the anticodon on a tRNA is UAC, and that tRNA carries the amino acid for methionine. If we were to look at that table that I gave you to help translate everything out, you would see that AUG translates to methionine, which is carried by that tRNA molecule. Right? So tRNA is kind of like the intermediary. It's if we were to actually write this out, right, us as humans doing the writing on the board, that's basically what the DNA molecule is doing. It's doing the actual translating itself. So then what we're left with is this sequence of amino acids in the appropriate order that's been read from our mRNA strand. I gave you this slide on Friday, but I decided not to go over the structure of a tRNA molecule. That's why I have a big X over it. You don't need to bother knowing this, all right? It will not be tested on, but I kept it in here because I did present it to you on Friday. So anything on this slide, not on the exam, right? We're not going over the structure of this molecule. But what we will go over is how this molecule is repeated, or how it's constantly used many different times. All right, we have our mRNA. We're translating it to a strand of amino acids. The tRNA molecule is the one carrying the amino acid and giving it away to our newly built polypeptide strand. Right, so if that tRNA is reading a codon and bringing in the correct amino acid and giving that amino acid away to the growing polypeptide, what's left is a tRNA molecule that no longer has an amino acid, right? It's giving it away to that growing protein. So somehow we have to bring an amino acid and attach it back to the appropriate tRNA so that molecule can be used again and again. That's where this really long named enzyme comes into play, right? This is our amino acetyl tRNA synthetase. So what it does, right, we have our enzyme here. This process uses energy, which is why we have our ATP molecule being utilized by this enzyme, right? That powers the enzyme to do this work. An amino acid will be in place, right? An amino acid will be in that enzyme. The enzyme is going to find a tRNA that's missing that amino acid. It's going to bind in the enzyme, and it's going to covalently attach the amino acid back to our tRNA molecule. So now we have a tRNA molecule that once again has an amino acid attached. It can once again start reading the mRNA. Right? So oftentimes we will say a tRNA that is missing an amino acid is called an uncharged tRNA. And the tRNA that has an amino acid, right, and it has the ability to give it away to help grow a polypeptide, is a charged tRNA. Also known as an amino acetyl tRNA. I think charged and uncharged is easier to remember. All right. Let's answer a question here. All right, I want to think about that enzyme. What would happen if the enzyme that put the amino acid on the tRNA molecules became defective? It no longer worked. What would that mean for the cells? Right, and talk with your neighbor, talk it out.
Wait, then mRNA wouldn't be able to find it.
So it's just making tRNA. That's not charged.
Or what if you produce the kind
Yeah. Oh.
Yes, I did think what I'm hearing from you is
I
correct.
don't know how to say it. Yeah, I don't know how to phrase it.
So basically, we have these amino acids.
Sorry, there's.
So what does that mean for the production of protein?
This is profit most.
No, it's just the big for cells.
Yeah, normally I have to push my
Tutors are Sunday through Thursday, 7 to 9, in Nobel on the 3rd floor.
Did you guys take 0.25 back?
There's a few.
Wow.
All right, so we have this enzyme responsible for putting an amino acid onto a tRNA molecule. What happens if this enzyme no longer works? What would be the result? What would that mean for the cell?
The tRNA wouldn't be charged.
Yeah, the tRNA wouldn't be charged. It wouldn't be able to carry an amino acid. So what does that mean about building a protein?
Not possible.
It's not possible, right? tRNA, maybe it can still read the mRNA, but it doesn't have an amino acid to give away, so it can't actually build the polypeptide. So that would be very bad, right? We need to build proteins in order to keep our cells working. All right, let's elaborate a little more. So if we look at this process with a little bit more detail, and yes, I did simplify these slides, so you know it's a lot of information, it's much less than original. We are using ribosomes, right? So we can't forget about the role of ribosomes in making a polypeptide. Now in prokaryotes, again, we're keeping it simple, we only need one type of ribosome. Eukaryotes, they're going to complicate that process a little more. They're going to have distinct ribosomes for different cellular components, right? But we're using ribosomes. The ribosome function stays the same, right? It helps to build new proteins. If we look at the structure of the ribosome, it's actually composed of two subunits. One is what we call the large subunit, one is the small subunit. Right, so this example here is kind of drawn out to how it would look, right, if we made like a computer model as close to the original thing as we could possibly get. The pink side here, this is part of the large subunit on top, and the blue part is part of the small subunit on the bottom. All right, now we're going to look at it in more of like a cartoon version. So it's going to look like this orange structure on the side, where the top is the larger portion. Up here you can kind of see the division between the top and the bottom on the sides here. So very distinct, right? But the bottom part is the part where the mRNA is actually kind of sliding through. All right. Now ribosomes, they are made up of rRNA. All right, so ribosomal RNA, right? So yet another type of RNA, right? We have our messenger RNA. We have our transfer RNA, which is our tRNA. Now we have our rRNA, which is what makes up the ribosome. If we look at the actual steps of translating mRNA into a protein, there are 3 steps, which are conveniently labeled in the exact same manner as the steps of transcription, right? Initiation, elongation, termination, right? So that should make it a little bit easier to study when you're studying for the exam here, both transcription and translation, they have the same names for the steps. Now, initiation is where we start reading the mRNA. Elongation is where we grow our polypeptide chain. And then termination is when we complete the entire process. All right, we will elaborate a little bit further into these steps. And so initiation, elongation, termination. We're going to start with initiation. Right. Oh, I always forget I put this image up here. I often think of a ribosome reading an mRNA strand. To me, it kind of looks like, if you've ever seen a tree start growing around like signs, it looks like the tree's eating like a pole. That's the mental image that comes into my mind. Maybe that'll help you remember as well. All right, but if we start with initiation, all right, the first thing is that our mRNA is going to bind to the small ribosomal subunit. Right, so the small ribosomal subunit is binding to our strand of mRNA there. Once it's bound, right, and it's going to be centered around the start codon, the start codon is going to be read, and what we call the initiator tRNA molecule will bind to the start codon. We call it the initiator tRNA because it contains the anticodon to the start codon. Right? It initiates the start of translation. So our tRNA molecule has come in, right? Its anticodon has found our codon AUG, and it's carrying with it the amino acid for methionine, which is coded by the codon AUG. Once that tRNA has come into place, now we're going to fully assemble our ribosome by bringing in our large ribosomal subunit. So that'll encapsulate the tRNA that was bound. All right, so small subunit attaches to mRNA. Initiator tRNA comes in, finds the start codon, and then the large subunit fully attaches to the ribosome. Now we're ready to actually read our strand. Right? Everything is in place to start. Right? Not only have we attached our ribosome, but we brought in the first amino acid to the sequence. Right? That start codon coded for methionine, which is our first amino acid. Our next step of elongation is basically just the RNA reading the mRNA in order and bringing in the appropriate amino acid. Right? So the aminoacetyl tRNA, which is also known as a charged tRNA, it has an amino acid on it, brings in the next amino acid in the sequence. Right? So in this image here, we are looking at elongation step, but kind of in the middle of the elongation step. Right? We've already started with our first one. We've added in a whole series of amino acids so far. So now we're going to look at how this polypeptide continues growing. All right, so we have our tRNA in this middle part of our ribosome containing the growing polypeptide. The next sequence downstream is being read by the next tRNA molecule, which brings in the appropriate anticodon, and it contains the appropriate amino acid. Once that is in place, right, that growing polypeptide is actually going to leave the tRNA that was in the middle and move over to the new amino acid. Now, remember that way back in Unit 1 here, we talked about covalent bonds, and the specific type of covalent bond between amino acids was called a peptide bond. Right? So we're covalently bonding these amino acids via these peptide bonds. Right? So the growing chain moves over to the new amino acid, forms a covalent peptide bond with it. And then the ribosome is going to shift and start reading the next sequence. Right? We call the action of moving the growing polypeptide to the next amino acid in the sequence polypeptidal transfer reaction, right? Just very literally says what is happening. We are transferring the polypeptide, right? So we're going from the growing polypeptide, transferring it to the new one, the next one in this sequence. And it's going to continue reading in that very same manner until it comes to a stop codon. Right, so we're just going to continue growing that polypeptide. tRNA comes in, right, with the new amino acid, growing polypeptide chain bonds to that new amino acid. The ribosome is going to shift over, allowing room for the next tRNA to come in, and so on and so forth. Now the last stage is termination. Right, and I realize I have a typo here. I've highlighted elongation on this side. I should be highlighting termination because this is the termination stage. So this RNA reads the mRNA, this ribosome reads the mRNA until it reaches a stop codon. Now remember, a stop codon does not code for an amino acid, but the stop codon still codes for something, right? And that something is a release factor. And it looks very similar to a tRNA, but it doesn't bring in an amino acid. Instead, it brings in a molecule, we call it the release factor, that basically tells the ribosome, we have reached the end, right? You are no longer needed. We have made the finished product. Everybody is going to start dissociating and breaking apart, right? So the finished polypeptide will leave. The ribosome will break apart and leave both of its subunits. And now we have our finished product. So I put in a video here to help us visually see what's happening here. So this video is going to show us the elongation and then termination steps. Right, we have our amino acid binding with the new amino acid that just arrived. We're shifting downstream. Chain is now binding to the new amino acid. It's shifting downstream. And just continues, right, until it reaches a stop codon. That release factor comes in. It doesn't bring in an amino acid, but it tells basically everything to start breaking apart and floating away from the mRNA, right? We are now done. All right, so I wanted to play it just one more time so you can see it over. Right, notice how the ribosome is shifting down. And that amino acid, the growing polypeptide chain, is always going to be covalently bond to the new one. Just kind of hops on over to the side as the new one comes in. Then that stop codon codes for that release factor, which says we are done with translation. All right, so big picture here. We're thinking about all of the central dogma, transcription and translation. What does it all mean, all right? Why do we need to know this? Well, if we think about DNA being transcribed to our RNA and then being translated to a protein, that protein has a function, right? And that function might be expressed in different ways, right? So in this example, we're saying that we're going from DNA to our RNA, and then we're translating this protein that relates to producing pigments of the fur of a mouse, right? So this forest mouse here, right, has the variation of this gene that calls for a dark coat. Versus this other mouse, this beach mouse, right, has a different allele that's being expressed, different version being expressed. And this version has a slightly different DNA sequence, right? It has this A at the beginning instead of the G, which leads to a slightly different mRNA strand being made, which leads to translation of a slightly different molecule, right? We have a different amino acid in the first part of our sequence here, right? But that's being expressed as a protein, still makes a pigment, but now that pigment is this lighter color. Right? So this is big picture. We're putting all the concepts together here. All right. Let's do a quick review here. And I realized in my last class, I already gave you the answers. So I want to make sure, talk with your neighbor, you understand how I arrived at the answers that I have provided you.
Well, they have two polys with one chain containing. Wait, how do you Oh, right, because it's the trick. Yeah, you divide, or you divide it into chunks.
Yeah, so amino acid is coded for by how many base pairs? Yeah, so for each amino acid, it has 3 nitrogen bases. Oh, maybe like pairs.
Yeah, that's right.
Yeah. All right, let's take a look at these answers to make sure we understand how we got there. So, insulin in both human and horses are the same length. They contain the same number of amino acids. Right? They're made up of 2 different chains. One chain has 21 amino acids. The other contains 30 amino acids. So, in order to go from amino acids to nitrogenous bases, right, an amino acid is coded for by a codon, and a codon consists of 3 nitrogenous bases. Right? So for each amino acid, there's 3 nitrogenous bases. I multiplied 21 and 30 respectively by 3 to get our numbers. Now, one thing that I did not do, and it was called to my attention in my last class, is that at the end of each mRNA, right, to tell us to stop translating, we have a stop codon. That stop codon has 3 nitrogenous bases, but does not code for an amino acid. So it might be more appropriate for this question to say how many nitrogenous bases are present in the DNA to code for chains containing 21 and 30 amino acids. Right? Then I would say, well, 21 amino acids is 63 bases, 30 amino acids is 60. But if I were to say, how many nitrogenous bases are in the DNA to code for each chain? Right? Well, I have to include my stop codon to tell translation to stop transcribing. So it would be 63 plus my 3 for my stop codon, 90 nitrogenous bases plus my 3 for my stop codon. All right, and then in what stage are both subunits of ribosome attached? That's in that very first stage, initiation. Small subunit comes in, our initiator tRNA molecule binds with the start codon, and then our large subunit comes in. Any questions? All right, we're going to move on to the last topic here, and again, I have really cut this lecture down to bare minimum. You'll notice that we're not going to talk about the consequences of mutations or how we're going to repair DNA from a mutation. We're just going to talk about the types of mutations that can occur. All right, so if we're thinking about mutations, we're thinking about changes in the nucleotide sequence, right? So somewhere along the line, right, we've got a mutation in our DNA. Maybe it changed an adenine to a guanine, right? An A to a G, or a T to a C, right? Something has been changed in that sequence. That will cause a mutation. Now the word mutation, when you associate like what happens with somebody that has a mutation, we often think of negative consequences, right? Well, that's not always going to be the case. It might have no effect at all. So a mutation might occur, but maybe that mutation still codes for the same amino acid. So even though there's a mutation, there's actually no change, right? Has no effect on the individual. It's a neutral effect. Likewise, that mutation, maybe it does cause a change, but that change is for the better. Whatever happened, right, caused the organism to survive and reproduce better than it did previously, right? That's beneficial. But on the other hand, we might have a mutation that is not beneficial, right? It caused something to happen that lowers the organism's ability to survive and reproduce. I have an example of one of these mutations in this image here. I have a normal fruit fly with its antennae, but a mutation had occurred that caused one of these antennae to now be a leg, right? So now it has a leg where an antenna should be. That's not very beneficial for this fruit fly, right? So this is not a good mutation, right? Or we can think about mutations, right? Our flower up here, maybe having one petal being a different color. Yes, it's a visible mutation, but maybe it has no effect on its ability to be pollinated by insects, right? So mutations, not always a bad thing. Let's look at the mutations, all right? So if we're looking at mutations in our genetic code, we can have point mutations. Point mutations is where a singular nucleotide is replaced by something, right? Or it's altered in some way. Now we're going to talk first about point mutations that are part of substitutions, where we are substituting or we are swapping out one nucleotide for a different nucleotide. One way that this can happen, I wouldn't expect you to know this for an exam, but it's something called spontaneous deamination, right? Where we can take, say, a cytosine through this process of spontaneous deamination, and it becomes thymine, right? That's an example of a mutation that has occurred. We've gone from a C to a T, right? In only one of our bases. Oh, don't know what happened there. All right. So let's look at some types of these substitutions, right? So point mutations happening at one nucleotide in our sequence. We're swapping it out for something, which means this is a substitution mutation. Our substitution mutations can have 4 different effects. We can have a silent mutation, which means, right, if we look at our original sequence up here, we might have a mutation. We're swapping out a base pair, right? So in this base pair up here, GGC becomes GGU, but that still codes for the same amino acid. So even though the nucleotide sequence is different, our resulting amino acid sequence is the same. Right? We would call this a silent mutation. We can also have a missense mutation, right? So this is where we have a codon that's being swapped out, And that result is that we are changing the amino acid. Right? So we've gone from GGC to AGC, which no longer codes for the amino acid, I think this is glycine. Now it codes for serine. Right? We've swapped out an amino acid. We can also have a nonsense mutation, which means we have changed a codon that normally codes for an amino acid into a codon that now codes for a stop codon. So what that means is that we are prematurely stopping translation of this mRNA, right? That protein is not being fully built because of this mutation. Right, so this one happened in the second codon here. We went from an AAG to a UAG, which means we're stopping. So in this sequence, we're starting with our start codon, and then immediately we're met with the stop codon. Not very helpful. Not a great mutation to have happen. Our last mutation, right, for a substitution, is a sense mutation. So this is where we have a codon that's normally a stop codon, but now it's coding for an amino acid. So basically what we've done is we removed the stop codon, and now the cell doesn't know, or now the ribosome doesn't know when to stop translating. Right? I don't have an actual image to accompany this one here, right? But we would just keep reading the mRNA with no stop codon in sight. So these are all types of substitution mutations. Right? I'm actually going to skip over this slide because we kind of talked about it already, right? In that a lot of times, say for serine here, the first two letters are the same, the third letter is different, so a mutation in the third letter will lead to the same amino acid. But for the sake of time, we're going to skip over it and move on to our next set of mutations, which are not substitution mutations. We would call them insertion or deletions. All right, where we're either adding in extra nucleotides or taking out some of the nucleotides. Right, so we're no longer swapping them. We're either completely deleting them or adding in a couple new ones. This can happen with one or more bases. It's often going to result in what we call a frame shift mutation. So it's going to change the way that we are reading the mRNA. So it's going to change what the codons look like. So let's look at an example here. Right? A frame shift mutation, we're either adding in or taking something out. If we were to pretend that this sentence, having 3 letters in each of the words, right, was a mRNA molecule, we would normally read it as the fat cat sat. But if we delete the T, that changes how we're grouping our codons, how we're grouping our sets of 3s. So now we have a sentence that makes absolutely no sense. Right? We're completely changing it around. So in the DNA sense, if we look at an actual example, right, we're going from a normal sequence to a sequence that has inserted a singular T. Now instead of deleting one, we're inserting a T. That changes how we read the rest of our mRNA. No longer are we reading CAG, CCC, ACT. Now we're reading CAG, TCC, CAC, and then we have this random one nucleotide T at the end. So that changes the amino acids in our sequence. Right, with that T at the end, really not coding for anything. This is different though if we add in a group of 3. If we add in a group of 3, we actually don't change how we're reading the codons. We're simply adding in another amino acid or taking out an amino acid. So in an insertion mutation with 3 inserted in, now we are just changing out one of the amino acids, but because there's a multiple of 3, the rest of the sequence is still read in the same way. Right? So it doesn't change how we read it. It does not lead to a frame shift mutation, even though it is still an insertion mutation. We're going to look at one more example, one more slide here before we move on to the content check. This is just reiterating this frame shift mutation idea, right? If we either add or delete in a base not in a multiple of 3, we're going to change the way that we read the rest of the reading frame, the rest of the mRNA. But if this happens in a multiple of 3, we either add 3 in or take 3 out. We're either going to add in an amino acid or take out a full amino acid, but the rest of the mRNA will be read as it normally would be. All right. We're not going to go over this slide together. I will make the answers available. I would encourage you to do this on your own because something like this is fair game for an exam, and you do have a full week to get these concepts down. And I did link some videos at the end. But I might ask you, what type of mutation is occurring? What is the name of it? And what is the consequence of this mutation? Does it change codon to read a different amino acid? Is it the same amino acid? Or is it causing a frame shift mutation? Right, so practice with this slide here. I'm going to move over to our content check, which I am going to give you extra time. If you don't get this done today, you can either submit it online by Wednesday. Remember, we don't have class Wednesday. Or you can put it in the mailbox next to my door if you have a physical copy. I want us to take a sequence of DNA, right, and transcribe it and translate it. Now I'm going to start you off together. All right. So if we look at our DNA sequence, right, we're going to use one of those strands to build our mRNA. Which strand is going to be read? It's the 3 to 5 strand, right? It's read 3 to 5 because RNA polymerase can only build in a 5 to 3 direction. So if we were to look at this, well, 3 prime, and then it's T, A, C, and then it continues on, right? This would be our template strand. This is the strand we are reading. Now the DNA polymerase is reading in this direction, but it's building in a 5 prime to 3 prime direction. So if we were to read this by DNA polymerase, right, T would be A would normally be T, but because it's RNA, it's a U, so that would be U, and C would be G, right, and so on. Right, so that would be our RNA. Now we would take our RNA, break it into groups of 3, use our codon table there, and translate this out to a protein. If you were to look up there, right, we would have the protein methionine because AUG is a start codon coding for methionine, right? And so this would be our amino acids. So what I want you to do is use the same principle to finish out this content check. If you don't turn it in today, just get it to me by Wednesday, either online or to my office door.